This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 212, recorded on 12/21/12. Well, ones and twos today. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Howdy there, Vincent. How are you?
I'm pretty well. How about you?
I'm just dandy.
You're dandy?
Yeah.
Do you have really dandy weather there?
Chilly by our standards, 55 degrees, 12.8 Celsius, clear blue skies, little breeze. It's okay. It's supposed to freeze tonight.
It's tough. It's tough.
I know.
We had some kind of heavy storm blowing through here last night and this morning. It was windy and raining. It's all cloudy, and it's still windy up here.
Wow.
You may hear the wind on my microphone. 6 degrees Celsius here.
It's windy in your office?
Well, the wind blows through the cracks in the windows.
Ooh.
So we're up here on the 13th floor, and you can hear it whistling.
That's disturbing.
So if you hear whistling during the this TWiV, it's not Rich, it's the wind.
Okay. I might be whistling. You never know.
Whistling Dixie?
Sure. Why not?
When you do that barbershop stuff, do you whistle sometimes?
No.
No.
I haven't done that.
Okay. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. How are you?
Also known as Lily Tomlin, right?
I don't know about that.
I bet you'd
Hi,
like
Kathy.
to be.
Hi, Rich.
You would like to be famous like she is, right? Oh, I'm sorry, you're famous already. That's right.
Well, yeah.
Forgot.
But
She's on
Yeah.
TWiV.
Yeah.
Yeah.
Forgot. And
yeah, somebody called me a rock star.
And Lily is not. Lily is not on TWiV.
Right.
She
How's
would
the-
like to be, though.
Who, Lily?
Yeah.
Send her an email, see if she wants to make a guest appearance. How's the weather out in Ann Arbor?
31 degrees Fahrenheit, -1 Celsius. Gray skies, but just a dusting of snow on the grass and trees.
It's going to be a white Christmas, huh?
Probably melt by then.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
And you must have gray skies, I bet.
Yeah, they were, and then it cleared up, and we've got a lot of wind. It's 56 Fahrenheit, 13 Celsius. But it has cleared up.
Did you have that storm this morning and last night?
Yeah. It blew through here, but, no major damage. It's not the end of the world. Oh, wait, it was. Sorry.
This morning, it blew down some power lines in our town. Can
just
you
what
believe?
you needed.
They just put them all back up, and a few were blown down right near the high school. Oh, man. Yeah. Well, yeah, the world is still here.
Yeah, it stubbornly persists.
And this episode is all ones and twos, right?
Right?
If we had recorded on 12/12/12, that would have been something.
Yeah.
Right? That was like a Wednesday or something like that.
Yeah.
But we got all 212, 12/12. No, today's the 21st.
12/21/12.
12/21/12. Of course, if we did it in a different calendar, it wouldn't be that way. But
Right.
and who knows? Maybe that'll work its way into our title. We'll have to see in the post-game show.
Yes. And that's the last time a date anything like that will roll around until a date something like that rolls around again.
Yeah, that's right.
Yeah.
That's profound.
Yeah.
Just like everything on TWiV. Dixon's not with us. He's down in New Zealand.
Wow.
Fishing, I suppose.
Or
And-
if the end of the world already came there, he's not.
Yeah, that's right. Well, if he doesn't show up in a couple of weeks, then... Because there's no other way I'd find out, I guess.
Is he visiting the Shire too?
He said he wasn't, no.
Oh, okay.
They do Hobbit tours down in New Zealand.
I would expect they would. That'd be
Yeah.
quite an opportunity.
Cash in, right?
Yeah.
Have you seen that movie?
No.
Yeah.
No, I found the book dreadfully boring, so.
How about the Lord of the Rings books? Did you like those?
Oh, loved those. Absolutely loved them, and I thought the movies were great, too.
But the Hobbit you didn't like, huh?
The Hobbit, it really didn't grab me.
Okay. let's see. We have a bit of... Are you there, Rich Condit? Did he go away?
Uh-oh.
Oh.
He's been unusually quiet. I bet he muted himself.
Maybe the end of the world happened already in Florida.
Ah.
Yeah, it could be.
Can you hear me now?
Yeah, and I figured if the world did end in Florida, they'd still be doing a recount on it anyway.
Yeah, that's right. Hanging chads and all. I was wondering why you guys weren't, listening to me.
All right, let's try and get started here. We have a couple of follow-ups. Rich, did you want to mention something about inaccessible TWiVs?
Inaccessible TWiVs? You mean things that people can't understand?
Yeah, exactly.
Right. Sure. I'll try and be brief. It occurred to me that we had been talking about metabolic labeling, a term which probably doesn't mean a lot to some people. And we were saying things like S35 and P32. We were talking about it in the context of labeling the VPG, the terminal protein on polio RNA. And I just wanted to very briefly go over that. When we say metabolic labeling is basically added radioactive tracer molecule to cells that are doing something so that it gets incorporated into biological molecules, and then you can, because they're radioactively labeled, you can follow them either with effectively a Geiger counter or a scintillation counter, or by exposing a film to proteins separated on a gel or something like that. And when we say P32, that's radioactive phosphorus, and that's a typical label that's used. And when you label with P32, you're going to label RNA and DNA, and in fact phosphoproteins and lipids, but you can separate all those things from each other and follow one or the other. When we say S35, the implication is that that's S35 methionine, which is an amino acid that gets incorporated into proteins, and so we can follow specific proteins. In the case of the polio and VPG, they labeled cells with S35 labeled methionine, and then I guess purified virus and the RNA and the virus with the VPG attached, and the VPG was then radioactively labeled with radioactive sulfur incorporated via methionine. So in the future, when we say stuff like that and you don't understand what's going on, interrupt us.
Yeah, I agree.
Listeners,
I think
okay.
And we should interrupt each other. I think we are getting highly technical, and we shouldn't.
Right.
And speaking of VPG, our next comment is from Richard Virgin Slane, who writes, "Awesome, you guys reviewed my paper. The hunt for VPG on linkase sure taught me a thing or two about protein biochemistry."
I'll bet.
Yep.
Yep.
Also an email from Robin, who writes, "An LPN, licensed practical nurse, sometimes interpreted as a low-paid nurse, is also known as an LVN, licensed vocational nurse. They do not have the same level of training as a registered nurse RN, a certified registered nurse practitioner, CRNA."
Wow.
Oh, man, complementary RNA has a substantial additional training past the RN. Wow, you thought virology had a lot of acronyms, abbreviations.
Well, I'm glad for that clarification, because I didn't understand the differences, and it's good to know.
Yeah, I think that came up in a listener email, right?
Yep.
Mm-hmm.
Yep.
Yeah.
All right. We are doing all email today. And let me start with the first one from Chris. "Hello, TWiV hosts and audience. Just wanted to share an interesting article with you." He sent a link to an article in DCIST, DCIST. Get it? Washington, D.C.?
DCist, yeah.
DCIST? I thought it was DCIST. That would
Well,
be
if they
good
stop
too.
publishing, that would be good, yeah.
National Park Service to start killing deer in Rock Creek this winter will give meat to food banks. Now, Rock Creek is the park by D.C., right?
Yep.
So I guess they have too many deer, and they're going to kill them off. Reminds me of Dixon's old diatribe on the problems of deer overpopulations and the expanding trichinella infestation in the early days of TWiV. Anyway, maybe some TWiV listener out there could arrange to get some samples from this. Seems like it could be a useful resource. Thanks for all the awesome podcasts, and thanks to the other listeners for the great and challenging questions. Totally makes my science life, and maybe my personal one too. Chris is in the Department of Micro and Immunology at Georgetown. Check, I guess that's why he knows about this.
That's probably, yes. Yeah. That's in the neighborhood.
Who had sent this? Wait a minute. They're going to use this. They're going to give this to food
banks.
Food banks. But you have to make sure it doesn't have prion diseases, right?
Yeah.
That was my first thought.
I remember somebody bringing that up somewhere else as well. But yeah, if you wanted to look at the virome of deer, that's a good opportunity, right?
Yeah.
Got to hook up with the hunters. I guess
maybe. These are urban deer. So it would be different from the virome that you might expect in a really wilderness area. Maybe.
It'll be interesting to find out, though.
Yeah, might be interesting to compare. And actually, this is not. This would be a good place to sample them because it's convenient to a lot of labs. But there are lots and lots of parks and residential areas around the country where they do stuff like this. Because in all the areas where we've eliminated the apex predators, which we tend to do. We have then often failed to take the responsibility of being the apex predator, so the deer overpopulate the area.
As you would expect, PETA is opposed to this.
Sure.
And they say when the animals are killed, it causes a spike in the food supply, which will only prompt the doe to breed at an accelerated rate. Did you know there was a feedback mechanism that said when there are more plants, there are more deer breeding?
That's why when they do population control like this, they usually go after the does. If it's done properly.
Right. Well, some lab in the D.C. area could take advantage of this. So
yes.
Thank you. Kathy, can you do the one from Jacqueline?
Yes, she writes, I wanted to email you about a program I found some time ago called Evernote. I apologize if this has already been recommended. I'm still trying to get caught up. I just listened to episode 45 where Dr. Dove recommended MediaWiki. This sounded complicated for those of us who are not tech savvy. Evernote, on the other hand, is a very simple program. You can create notes of any length, attach files, create tags, and sort things in notebooks. In addition, there is a very nice search feature that will find particular words within notes. I've been using this since I started grad school last year and have found it invaluable. The one caveat to the program is that there is a maximum you can add to it per month. For the casual user, this will not likely be reached. However, for a nominal fee per year, that maximum is drastically increased. And there does not seem to be a lifetime limit. You can either download the program on your computer or interface with the program on the internet. In addition, you can sync your notes between multiple computers and access them anywhere there is internet. I use this program on my iPad, iPhone, and my PC at home, so the program works on multiple platforms. Thank you again for the great podcast, as it kept me busy during my 20-plus hours of travel this previous holiday weekend.
I think this might be the July 4th holiday weekend.
Oh.
Right. Okay. Wow. We're that far behind.
Episode 45 is a long time ago.
Yeah.
Actually, I think either I or somebody else had Evernote as a pick of the week at some point. Mm-hmm. And just a little footnote on the MediaWiki thing. I have moved on from that and have since adopted a different system for keeping track of stuff because it did turn out to be a lot of management overhead.
I use Evernote to save, like if I come across a website, it's a story for TWiV or TWIP or TWIM, I save it in Evernote. And I have 3 different folders in Evernote, TWiV, TWIP, and TWIM. You can put it in there and get back to it on any computer, so it's kind of nice for that. One of my students keeps his entire lab notebook on Evernote. It's very nice.
And backs it up, right? So it's
he's backed up all over the place, yeah. But you can put data, text, gels, PDFs. It's very, very handy. He likes it a lot.
So just along that line, when I got my iPad, somebody pointed out to me that there's an application called AppStart. And at the time it was free. I'm not sure if it still is. But they consolidate and have short little reviews and then links with the prices indicated for lots of categories. So there's a whole category called Notepad. There's one called Personal Assistant, Office, Newsstand, Reference. There's pages and pages of these games. And so you can compare 6 or 7 or 10 different types of note-taking applications or cooking applications or music applications. So App Start, and it also works for iPhone apps, and I don't know about Android apps, but that's how I found out about
that's
Evernote.
That's good.
That sounds very useful.
Yeah.
Okay.
I use the Notes, just the very simple Notes thing that's built into the Mac OS X.
Mm-hmm.
But that's not sophisticated or I wouldn't keep a lab notebook on it or anything.
All right, Alan, can you read Edwards, please?
Sure. Hello, Twiverers. Thank you for the entire stable of TWiV podcasts, which have turned the cookery parts of my bench work into a chance for wider reading. In TWiV 210, Mike wrote about asking whether GM chickens and pigs, that's genetically modified, not General Motors, chickens and pigs lacking sialic acid receptors could be introduced, reducing the number of domestic animals able to harbor influenza. As you may have seen, something similar has already been demonstrated in principle. GM poultry and pigs have been around in labs for a long time, many of them using GFP as a marker, which leads to an amazing selection of glow-in-the-dark animals, surely a missed commercial opportunity in itself. In 2011, Lawrence Tilley and colleagues showed the same technology could be used to introduce shRNA with anti-influenza activity, and he provides a reference. Although it's not really discussed in the paper, I gather that the small number of progenitors used to rear the world's current massive numbers of broiler chickens means that the transgene of this sort could be introduced very quickly if there was the will to do so. However, as you commented, just because you can do something doesn't mean that there's any desire to do it. And at the moment, the public's concerns about GMO, both considered and reflexive, clearly outweigh the fear of globally disseminated avian flu reservoirs. Let's hope this possibility doesn't become more alarming in years to come. But if it does, it's good to think that we may have the technology to intervene. Thanks again for the podcast and best wishes. And Ed is at Worcester College at University of Oxford.
Yeah, this story he's talking about, we talked about on TWiV 116, cocaine colonies and chickens. They had introduced siRNAs into chickens and against avian influenza, and I think they affected spread.
Now, glowing antiviral chickens, I'm sure that would be a big PR hit.
It turns out that there's a website called Glowfish, and you can get fluorescently labeled fish. So green.
In the pet trade, you mean?
Yeah. Cosmic
Yeah,
blue, galactic purple, starfire red, sunburst orange, and electric green. Yep. I'll put the notes here, and you can include them.
And I guess you just put a black light on your tank or something.
Oh yeah. So maybe in the FAQ they talk about how do I see these colors.
You can see them in you can see them in invisible light. There's enough fluorescence that you can see it.
Okay.
Yeah, I was just thinking to intensify it.
You could intensify it.
Right. Right.
Now this in 210 they were saying, can you make animals without sialic acids? And so that's just not going to happen because you need sialic acids.
Right.
But you certainly can modify them, as shown by that paper, yes. All right. Let me just read these two quickly because they mainly have to do with me. Ricardo writes, just a word of friendship. Hope everything is okay with all of you after Sandy. He addressed that to TWiV fellows. And Ricardo's our friend in Portugal. And Lance writes, just looking at all the pictures of Sandy on the web, hope you're all okay. And so you've probably heard that we are, because we've had a few episodes since then. But I just wanted to thank people for thinking about us. It's nice. Appreciate it.
Everything's sorted out. You're back to normal,
No.
more or less?
No.
No?
No, our shore house is all screwed up. But
Right.
you just can't go and fix it because the island is still closed. And I mean, there's no electricity on our house. There's no gas. The circuits are all messed up. It's going to be a bit of work. I'm not worrying about it right now. Cool.
Sorry about that.
What was that?
It was some sort of website of the thousands that I have up here, preparing for this episode, and some video went off.
All right.
I
Well,
don't know what it was.
in reward, you get the next email, Rich, from Peter.
Okay, dear Twivers. Peter writes, Hi TWivers. There was an interesting article in our local media today about a Hendra virus vaccine for horses. I thought it was unusual in the level of technical detail that it included. Thanks to listening to TWiV, I understood a little more of the background to the technicalities than I would otherwise. One interesting point the article makes is the use of a cross-disciplinary team to develop the vaccine. That made me consider that your coverage is centered on straight virology papers, and I have not heard a lot of discussion about such teams. Do the presenters have much experience with such teams? Are there interesting stories about how the different specialties bring pieces to a solution? Just thought it might generate a bit of a different discussion. Plus, I'm sure there are some interesting finer details about bats, horses, humans, and Hendra. Thanks for the podcasts. Pete, and Pete's in Sydney. And I think we've discussed this vaccine for horses in the past, but he links to an article in the Brisbane Time that talks about the development of... See, it's the same thing.
That's that video, yes.
It's the ad on that website. I got rid of it. Okay. He links to an article in the Brisbane... What was it? The Brisbane Times.
Well, go ahead and open it again, Rich.
No thanks. About a team of scientists that developed a subunit vaccine that's based on the Hendra glycoprotein that can be used in horses, and the idea is to stop the transmission by stopping it in horses. It's easier to get a vaccine, a veterinary vaccine, through the regulatory agencies than a human vaccine, and if you can stop the transmission to the horses, the humans who mostly catch it from the horses are protected indirectly. So,
right.
In that article, they spoke with Middleton, D. Middleton. I forgot her first name. Deborah. So her latest paper is in 2011. A recombinant Hendra virus G glycoprotein-based subunit vaccine protects ferrets from lethal Hendra challenge. So this is, I guess, expressing G, the glycoprotein of the virus, just by itself. I think they purify it, and then they inoculate that into ferrets, or in this case, horses, of course, and it's protective. So as Rich said, you can do that. If you want to protect ferrets or horses, it's a lot easier than people.
Yeah.
Multidisciplinary teams. Yeah, I guess we don't really note that. Although we do note sometimes when there are a lot of different groups and they're in different places internationally, and we remark about that. But I would say most of us in our careers have mainly worked, if anything, with a few collaborators, but mainly on our own.
Right.
Mm-hmm. But a substantial number of the papers that we've talked about on TWiV have actually been from multidisciplinary teams. We just haven't really called that out.
Right.
Because that's the way, particularly for something like this, where you have a very translational research type of goal, as they would say in industry, where you want to actually put a vaccine in a bottle. You're going to need people with a wide range of expertise, and that's not going to be in one lab.
Yep. Mm-hmm. All right. I'll take the next one from Adrian. Dear Professor Racaniello, this is Adrian from Singapore, where the temperature is 25 degrees Celsius with 94% humidity.
Wow.
It's warm, isn't it?
It's moist.
It's kind of room temperature heat, but it's very wet.
I've heard about Hurricane Sandy and was following your Twitter and hope that you and your family are fine. Thank you. I'm a recent PhD graduate from the National University of Singapore, from Professor Paul McGarry's lab, working on antigen processing of Epstein-Barr virus, but used to work on dengue and enterovirus 71 before. Now I'm looking for a postdoc somewhere. My wife introduced me to TWiV, and I'm a recent convert. I've been trying to catch up with the past podcast. You guys are great. I really enjoy the discussions about great papers and the Virology 101. Please keep up this great service. We visited your TWiV Facebook page, and we saw Reyes plaque fort, and we thought finally someone else out there understands us, as we also have our collection of plaque plates, as we did our fair share of plaque assays with both dengue and entero 71. Cool. Best regards, Adrian, Singapore. Okay. And there's a little coda which we'll maybe mention on another episode after the first of the year. Okay, Kathy, can you take the next one?
This is from Steve, who's been doing transcribing for us. So, if and when it seemed appropriate, it would be nice to hear what TWiV thinks about what disruption the new online education model may have on the operation of universities. By the initial quality offerings by Coursera and others, it seems that online models could eventually displace some, even much, of on-site, non-hands-on courses delivered at most academic institutions. Do you think the growth of quality online information and training will change the role of universities? If the ability to take good quality academic training at reasonable costs online challenges and begins to displace significant portions of the current mass undergraduate training model, how will, could, and should institutions respond? How should they redefine themselves? What could be the benefits and the dangers in shifting to more accredited academics online? More importantly, if shift and disruption happens, how should basic research science view this disruption and position itself to grow and prosper during this time? How can the academic science system adapt to embrace the change and feed more minds and create more social and political awareness of the deep benefits and fascinations in science? If the model changes, how might this change affect how basic academic science is done and or funded? There's a lot of questions that could be asked. I think you just asked them all.
Yes.
From will it change to if it changes, what will change to what are the core services and functions the current university model gives to basic research that it might lose with change, to are there good alternative models to the current system and what elements of any alternative should the academic science system try to adopt now and under conditions of change?
That's a passel of questions.
Yep.
Yeah.
So I have a couple of ideas because we're doing this here at Columbia.
Mm-hmm.
So Coursera is this MOOC, M-O-O-C, massive online, I forgot what the other, something course. Massively, anyway, it's M-O-O-C. A lot of people can register for your courses. It was started by 2 Stanford graduates, and the quality is mixed, but if you go through a course and take everything, you get a little certificate from the course person. So Columbia just joined Coursera in the past few months. So they're using my virology course next semester as a model to see how this goes. So I went down and I spoke with the dean this past week, and this guy is actually a journalism professor who was now moved over to the dean's office to manage the digital future for Columbia courses. All right. So nobody's making money on this. Columbia's not making money. Coursera doesn't have a money model, so I don't know how that's going to work. But basically, I take all my recorded lectures and put them on Coursera. I add some tests and quizzes, and people can get it. So Columbia's very excited about this. They feel that it will make their university more compelling to attend because, 100,000 people can take this course, but they can't all attend Columbia. In fact, Columbia College only has 1,000 students. So they don't view it as a disruption. For science, my view is what makes a great university is to have great research people together because it goes beyond science as well, right? So I teach a course because I'm here at a university and I benefit from all my colleagues and all the interactions and seminars and so forth. So I don't know if you're going to switch to an online model, you could get rid of the classrooms, but you still need to have all these professors together to interact and make a great university, right? So I don't know how you get rid of that. You can't have people sitting at home making great courses because at least for science, that doesn't work.
I agree. I don't think anything ultimately is going to completely replace FaceTime. So the most important interactions I have are just, having Dave Bloom in the office next to me.
That's the original version of FaceTime, not the app.
Yeah, right. Just stuff that happens. And of course, then nothing can replace people working together in a laboratory. You're not going to do that digitally. So I think you can build digital pipelines into that stuff so that other people can observe and take advantage of some of it in the fashion that you described the Columbia course. But I don't think anything's ever going to replace actually having people together interacting directly with each other. At least I hope not.
I've been recording my courses for the last 3 years and putting them online, right? So the students can listen. And every year, about half of the registrants show up and half don't, right? Which is fine with me. But those are the same 50% every week who show up. And they say they want to be there. They want to be able to ask questions and look at us and interact with us. And I don't know why that would change, right? That's basic human nature.
Yeah. Yeah, I'm reminded of the New Yorker cartoon from a long time ago where the students in the audience all aren't there, but their tape recorders are. And then up at the front, instead of the lecturer, there's a tape recorder playing the lecture. At least I think it was New Yorker. But what I started thinking about was the parallels in, how the music industry has changed because of the internet and how the publishing industry is changing because of the internet. And I don't think we can completely foretell where this could go. But I think Steve raises some really interesting questions.
I think there's a general realization that nobody's going to make money selling information anymore. If that was your business model, as it was, or at least that's the way it was thought of in the newspaper industry, then you're going to go the way of the newspaper industry.
Yeah.
So I think universities have very intelligently reached the conclusion, hey, we don't sell information. We can't. Let's put the information out there for free. And really, they've never been in the information selling business. It's always been possible to get all the information that you would get in an Ivy League education by spending a lot of time at your local library. Right? This has been available. That's not why you go to a university. You go to a university, now this is from the student's perspective and the undergraduate student's perspective, for example, would be that they're going to a university for the experience of college and for the direct connections that they'll get with the professors and with the other students and with colleagues in that environment. So what universities are selling is a particular environment which you're not going to put online.
Yeah.
Right.
And that's kind of what you said about the research side, too.
Yeah. So MOOC stands for Massive Open Online Course.
Thank you.
And Michigan is also part of Coursera. It looks like there's about 8 Michigan courses that are listed currently.
So I think what this whole online course does is just gives the information to more people who couldn't get it otherwise. And I think we love that here on TWiV, right? That's what we do.
Right.
And it's just a perfect extension. So I think everyone should listen to as many courses as they want. They're not going to replace the university, as Alan said, for a lot of reasons.
Yeah, my cousin teaches an English course completely online, so she does it from home. And she said there's a lot of veterans that take the course. And so you could imagine that might be a good venue for people that, may have some physical limitations about getting around. They can just take some courses online.
Sure.
That sort of thing. Or anyway, people just fitting it into their schedule. They have families and that sort of thing. So,
there are operations. I won't call them universities, but there are operations online that profess to be universities, and I'm sure you've seen pop-up ads and spam from them. Where that's the whole model, is that you take lectures online, and I guess you do exams, and and they are regarded more or less as diploma mills. A degree from one of those is not worth the electrons it's printed with, probably, but there are a lot of people who are paying for that sort of thing, and I think traditional universities can do a tremendous public service by stepping in and saying, you don't have to pay for that. You can get Vincent Racaniello or Kathy Spindler or Rich Condit lecturing on virology. Why are you gonna pay to hear Joe Schmo when you can get the lectures for free? And the point is that the lectures are not the college.
Yep.
Right.
Well, my course from this past spring is on iTunes, my virology course, and so far I have 70,883 students.
That's just amazing.
Isn't that incredible? So basically, they click and subscribe into my device. That's what that means. But it's really cool that I can reach that many people and spread the word. All right. I'm sure we'll come back to this many more times.
Yes.
We should get that Dean up here and tell us what he's thinking about, too. That would be cool.
Yeah.
Are you next, Alan? I don't remember now.
I think I may be next.
All right.
Taylor writes, Vincent and friends, I work at Children's Medical Center in Dallas. I'm a huge fan of your podcast, and so are all the students working here in credentialing. I follow a number of podcasts, and I recently listened to one called Science Sort of. I think it would be interesting if you and your co-hosts listened and critiqued the information here and generally made fun of it whenever possible. Just an idea. I think it would be ridiculously entertaining to hear you guys add your 2 cents, shoot down falsities, and show your own listeners the broad spectrum of questions that can be answered by looking at things under the microscope. So often, podcasts regarding science and technology can be pandering to the novice or only covering general information. You guys are keeping things real on TWiV, and you're seriously doing the Lord's work. Keep it up. Is that Lord Grantham?
I'd never heard of Science Sort of, but it seems to be a podcast and with 5 or 6 scientists in different areas, none of them microbiology. And I went through, I didn't find anything that we could really pick apart. So I'm sorry we can't do that. Didn't see any microbiology on it.
No, these guys are astrophysicists and paleontologists, all well-credentialed individuals. And I listened to a little of it. It's, they pick interesting current stuff and are mildly irreverent about it.
Mm-hmm.
It's an interesting little ditty.
Mm-hmm.
I'll keep my eye on it and see if any microbiology stuff comes up, but I have a feeling they'll stay away from it. It's not in their areas, right?
It's been around for a while. They're on episode 159.
All right. Mm-hmm. All right. We up to Rich, is that right?
Beats me. I can't, with the geography the way it is, I can't even keep track of whether we're going clockwise or counterclockwise or...
well, why don't you go? It's fine.
All right. This is kind of counterclockwise.
Darren writes... we should go clockwise. That would be good. All right.
Yeah.
So that means Kathy's next.
Kathy's next.
All right. Good. It's a great idea. That gives it it gives it a meme. That's perfect.
Right.
Thank you.
So am I doing Darren right now?
No,
sure.
Okay, sure.
Darren writes, Hi, everyone. I just want to say thanks for your podcast. Over the last few days, I've been listening to the episodes at work, and I've really enjoyed them. The show is awesome. I really appreciate it. Again, saying thanks and keep up the good work, Darren. And then he has...
Darren follows with a disclaimer.
Disclaimer.
Longest disclaimer I've ever heard.
I just before you do, Darren, I found this interesting because Darren is a financial analyst, I guess.
Yes.
So he's sitting at work, listening to TWiV. I think that's great.
Yeah.
And he has the disclosure is longer than his email.
Far, far longer than the email.
Right.
Alan, could you read one paragraph really fast?
Invitation to consider a derivatives transaction. This communication, where prepared by the sales and trading desk or desk strategist, may be marketing material, desk strategy, and or trader commentary. It is not a product of the research department. This material may constitute an invitation to consider entering into a derivatives transaction under U.S. CFTC regulations, sections 1.71 and 23.605, where applicable, but is not a binding offer to buy or sell any financial instrument. The views of the author may differ from the others at Royal Bank of Scotland PLC, the Royal Bank of Scotland NV, and or RBS Securities Inc., collectively RBS.
Wow. We'll play
Excellent.
2x. We'll play it at 2x speed.
Yeah.
Anyway, that's cool. All right, we're going.
The next paragraph is 3 times longer than that, and then there's a USA Patriot Act customer notice at the bottom. So,
wow. For thanking us to do TWiV.
Yes, and we have now been thanked and warned.
Thank you, Darren.
Thank you, Darren.
So, since we are going clockwise, that would make Alan next with Manuel's email, I believe.
Wait, are we going clockwise or counterclockwise?
We're going clockwise.
Well, you're north of me on the clock, right?
Yes, I'm north of you on the clock.
Manuel.
Manuel writes, Dear TWiV, he provides a link to a Wall Street Journal article, which he then quotes, a cure for color blindness might even be in the offing. Vision scientist Jay Neitz and his colleagues at the University of Washington are building on their 2009 breakthrough in which they restored red-green vision in 2 colorblind squirrel monkeys by inserting the missing gene into a virus and injecting it into their retinas. 4 years later, the monkeys, Sam and Dalton, still pass daily vision tests, identifying colors on a computer screen correctly. They also have a newfound liking for green M&Ms, Dr. Neitz says.
That's how they see whether their color's restored, I suppose,
Yes.
right? Interesting.
I've always loved the colorblind tests. You know, they're really interesting. And this article is the Wall Street Journal article is cool because they've got these little demonstrations of what it's like to see things with certain specific colorblind defects. I think those are cool too. So the original paper is a Nature paper, and it's actually co-authored by 2 people from here at UF because they use an adeno-associated virus vector to do this with. And the adeno-associated virus as a gene therapy vector was actually developed here at UF, and these scientists in particular, Bill Houseworth, who is actually he was originally in my department, but he's actually in ophthalmology now due to some sort of political quirkiness long time ago. At any rate, they also developed this technique for injecting this vector into eyes for a variety of eye diseases, most notably retinitis pigmentosa. They can actually help people see again. It's really pretty remarkable.
So in this case, the gene is L-opsin.
Mm-hmm.
And that helps. I guess that corrects color blindness, right? Red-green anyway.
It's a pigment.
Yeah. So when I visited you, Rich, I met with Nick Musiczka.
Right.
And he told me this story because he's worked on the vectors, right, as well?
Yes, he's primarily responsible for developing the AAV as a vector.
Yeah.
I think he's the primary person who deserves the credit for that. And he's continued to do that. He was the original director of the Powell Gene Therapy Center here, and he continues to work on development of AAV as a vector, and he's also done a lot of the basic science with AAV as well. So that's a big deal around here.
We should probably point out that the reason a lot of this is going on with eye diseases is the eye is immunologically privileged. So you don't have to worry as much about has the patient ever been exposed to this virus before? Are they going to mount an antibody response against it if you're putting it directly into the eye?
And AAV persists, right? So that's all.
Yeah, there's a whole string of ironies with this that are really sort of interesting because one of the reasons that AAV was originally chosen, I think one of the rationales is that it actually does a site-specific integration. The wild-type virus does a site-specific integration into a fairly innocuous region of the chromosome, somewhere on chromosome 19. And so the thinking was that you could use this to actually integrate a transgene into the genome and have it expressed indefinitely, and you would know exactly where it went so that you didn't have to worry about side effects. Well, it turns out that when you take the guts out of the virus to put in a transgene, then this site-specific integration goes away. And that was a little disturbing for a while until they realized that the vector persists as an extra chromosomal element, basically a plasmid, indefinitely. Which is almost better than integrating, and you get gene expression over a very long period of time. So it's been an interesting road. We probably ought to do one of these papers.
Yeah, I was just thinking
detail.
We have not. Yeah. All right, the next one is from Paul. I saw your Penn and Teller video on vaccination on the TWiV page. Listener pick TWiV 202, apparently advocating the acceptability of a 1% autism rate. They failed to mention that a 1% fatality rate from variolation was considered highly risky, even in the 17th century. And he provides a link to an NIH site about variolation, sparing the zeal for and popularity of a truer prophylactic cowpox. So the clarity of their point defeats me a little. Other than the above, I have no opinion on the acceptability or otherwise of vaccination. All right, well, this was a listener pick on TWiV 202, and they didn't advocate the acceptability of a 1% autism rate. What they said was, even if vaccines caused autism, and then they said they do not, and they used profanity to emphasize that, it would only be 1%. And I also thought that was a weak addition to the otherwise good video.
Yeah,
I don't think they should have said that.
I agree.
They should have just said it doesn't cause autism. End of story.
Yeah, it does. It does. It almost makes it sound as if a 1% autism rate is something that's acceptable.
Right.
Okay.
Right.
And that's a little. Of course, I was going to say it's a little harsh, but they're a little harsh.
Yes,
they are harsh.
Yes, they are deliberately trying to make a very strong point.
Right.
Yeah.
And I would take just to note to Paul that comparing the autism rate to a fatality rate is probably not going to sit well with a lot of people either.
Right. But the point is that, variolation had a 1% fatality rate, but smallpox had a 30%. So it was considered a good trade-off for the time, right?
Mm-hmm. For the time.
And of course, then Jenner did his stuff, of course, and because it wasn't acceptable. But that's how science proceeds. You just do better things. I find it weird that you have no opinion on the acceptability of vaccination.
Yeah.
We all do.
Yes,
Yep.
we do. Do it.
I think we've made that pretty clear over the various
All
days.
right. Moving clockwise around the United States, must be Rich Condit's turn next.
Sarah writes, Dear Team TWiV, sorry if this question has already been posted to TWiV, as I'm just catching up in the last few months. I remember a while back hearing a story that FDR may not have actually contracted poliomyelitis, but rather Guillain-Barré syndrome. I haven't heard much mention of this since and was wondering what, with your knowledge, collective knowledge about poliovirus, you might have good insight into this. Love your show. It makes the hours spent on plaque assays fly by. Keep up the good work, Sarah. I can't provide anything further into this. Can you, Vincent? Do you know anything about this?
Well, as everyone knows, FDR was thought to have polio. And, he started the National Foundation for Infantile Paralysis and raised money and made the 2 polio vaccines, funding their research. And then a while ago, someone said they did an analysis, of his symptoms. And I'll link to a page on this. It was published in 2003. So, 6 of his 8 symptoms favored Guillain-Barré over polio. So, for example, ascending paralysis for 10 to 13 days, facial paralysis, bladder bowel dysfunction for 2 weeks, numbness, lack of meningismus, and descending recovery. Those are all typical of Guillain-Barré. And 2 of his symptoms favored polio, fever, and permanent paralysis. So I think he had polio because Guillain-Barré typically doesn't give you permanent paralysis, but I think it can. There are always exceptions. But the point is, we'll never find out, right? So I like to say he had polio and supported the development of 2 polio vaccines.
When did he contract his illness? Was it
after a day of swimming, wasn't it?
Well, somewhere he was up at his hot springs place.
Age 39.
Yeah.
During his vacation in Canada.
Okay.
Campobello, not the hot springs in
Campobello,
Georgia.
that's it. Right.
What year?
1921.
So that's the real peak of the epidemic was like in the 50s. Is that right?
40s and 50s, yeah.
But certainly right after swimming makes sense.
It says there were no cases reported on or near the island, but that's okay because, as we know, there are lots of inapparent infections.
Right.
So Marinos Delakas, chief of NINDS, called the study a significant stretch, stating that Roosevelt's reported symptoms strongly suggested polio. I know Marinos, he works on polio. He's a neurologist.
All right.
Let's see. Well, anyway, he had a spinal tap, but it's gone. He had polio, I think. It's just an interesting idea to think about it, but I think his symptoms were polio, for what it's worth. Marinos thinks it's polio, so that's good enough for me.
Yeah, it's very consistent with polio, and it's certainly the historical significance of it is that he believed he had polio.
If he thought he had Guillain-Barré, he would never have made a polio vaccine, right?
Right.
Okay. Did I just do that email? No, that was Rich. So
that was
that's
Rich.
Kathy's next with Jim.
So Jim writes, Vincent, I just read about Learn Street in an uncollege newsletter. Went there and started using it. I don't code. I hear it's an important skill. Made furtive starts at Code Academy, and am impressed enough with the introduction and beginning steps at Learn Street to pass on the info. Your students and perhaps some of your listeners might have a need. FYI, in case you haven't seen it already, this Quora topic on skills researchers should have to communicate caught my eye. Hope you and your workplace have made it through the storms okay. Regards, Jim. So it turns out I got faked out. I looked at Jim's later letter links, but I didn't look at either of these, so I need you guys to help.
I got a beach ball. I can't do anything. I went to Quora, and it screwed everything up.
Oh,
so
right.
Learn Street is one of these sites. Code Academy is another. I think there are a few others that are aimed at teaching computer programming to people who are not computer science majors. And there's been this has become a very popular thing to do. I know a lot of science journalists who've decided to do this. I think this past year there was the Year of Code or something like that, where a bunch of people were going to try and learn programming, however you interpret that. I don't know. As somebody who has actually taught himself a few programming languages, I would say if you want to learn programming for the intellectual stimulation that comes with it, which is substantial, and it's a neat way of looking at things and thinking about the way stuff can work, go for it. If you think that it's going to be an important job skill and you're not already hired to be a programmer, you're going to be disappointed. Mm-hmm. It's not something that you actually need to be able to do in the modern world, despite what you've been told by places like Learn Street and Code Academy. It is a totally worthwhile thing to do. It's absolutely fascinating to learn, and I highly recommend it. But just go in with the expectation that this is like learning to play a musical instrument. It's something that you're doing for you. It's not something that you're doing to boost your paycheck.
All right.
And that's
my take.
This other thing of his. The Quora, the skills you need to have to communicate in academia is really pretty good. It's got this little table here that talks about various scenarios for communication, how long it takes, what your audience size is, interactivity, and visual aids. And it's humorous. It's pretty good.
All right. Thanks, Jim. I believe that brings us to you, Alan.
Sure. Faruku writes, Dear Sir, I am just curious to ask what links the TH1 and TH2 responses.
We never talked about them, have we, on TWiV?
I think, well, we've mentioned them obliquely, haven't we?
I don't know what it would be in connection with, but I can imagine, sure.
Right. Right. So I guess you or somebody else posted this very succinct summary that virgin helper T cells bear CD4 and T cell receptors, and depending on what signals they get from dendritic cells, they make either Th1 cytokines to help a killer T cell response, or Th2 cytokines to help a B cell response to mature into antibody-producing plasma cells.
I mean, by link, I suppose that he wanted to know how they were similar, right? I wasn't really quite sure about.
Yeah, I mean, those are the two arms of it.
Yeah, that's what I
Well,
know.
and also that Th1 type cells make things that promote more Th1ness and inhibit Th2
Yeah.
and vice versa. So that kind of links them.
Yeah, it's a yin-yang sort of thing.
Yeah.
All right. Rich Condit,
Am I up?
you're up. No, is it me? I'm
No.
sorry. We're going from Massachusetts to New York. Gee, I can't get it straight. Tony writes, Dear TWiV, I'm an aspiring molecular virologist. I listened to the interview Professor Vincent had with Professor Philip Marcus about cloning HeLa cells, TWiV 197. I couldn't understand, though, the part where Professor Marcus refers to the situation where his supervisors thought they were converting a gram-positive to a gram-negative, and he advised them otherwise, which led him to losing his place. Could you please elaborate on what happened in this situation, as I couldn't get my head around it? Anthony is from King's College, London. It's funny. So basically, they were testing a method of decontaminating, I think with polyethylene glycol, if I remember. And they would put E. coli into this room, and then they would decontaminate it, and then they would take samples and see if the E. coli would grow out. And they found that with time, E. coli became, which is gram-negative, became a gram-positive. And they said, oh, my goodness, we're converting a gram-negative to a gram-positive. Or maybe he said gram-positive to negative, but I think it was going from E. coli to a gram-positive. Mm-hmm. And so he said, well, what you're doing is you got spores in there of a gram-positive, and they're sporulating when you do your sampling. You're not changing E. coli into something else. And they said, oh, really? And they fired him.
Right. Because they didn't want to hear that.
No. It's probably a good thing that happened to him. It was so funny when he talked about it, though. So that's what happened. It was just a contamination with spores of a gram-positive, like probably Bacillus subtilis, common spore-forming gram-positive organism.
Yeah, and they were evidently annoyed enough at him for either figuring this out or whatever that they fired him. That is kind of surprising.
Yeah, he doesn't want to work at a place like that.
Right.
Nobody does, right? And you're right, Kathy. I had forgotten about the transcript. That's great to be able to go back.
It's transcribed. This particular one was transcribed by Rafael Fernandez.
Yeah.
And so it was very easy to go to it and go right to the place where Philip Marcus said that. So that was nice.
Yeah, that is handy to have the transcripts.
That's great.
There's been several times when I've tried to search audios to find out where something was said, and the transcripts would be a much easier way to do it.
Yep.
Or even searching back shows. I know we mentioned this on a TWiV, and if it wasn't in the show notes on the website, then it doesn't come up.
Yep.
All right.
Now it's my turn.
Now it's your turn.
Yes.
Okay, Rich.
David writes, Hi, sorry for the shameless self-promotion, but I thought our latest paper might be of interest to TWiV. We show how to combine deep sequencing and high-throughput proteomics to study adenovirus infection of human cells. Why? Well, two things seemed useful to us. Firstly, we demonstrate that this approach correctly correlates and identifies all the detectable proteins and transcripts present in this well-understood system. This means it will work even if you do not know that we were working on human cells infected with human adenovirus. In principle, you can look at any new virus in any eukaryotic host, mammalian, plant, or insect, and gain insights into changes in gene and protein changes on a very large scale and instantly identify pretty much all of the proteins detectable by mass spectrometry and collect them and connect them to their transcripts, both viral and host cell. So you can use this approach with any virus in any cell type, even if the host organism or the host virus has not had their genome sequenced or properly annotated. Secondly, we showed that by monitoring changes in gene expression and correlating that with protein levels, you can focus in on proteins that are targeted for degradation by virally induced mechanisms, i.e. the virus deliberately has the protein destroyed because it gets in the way of viral replication or spread. We searched for proteins that had been degraded during viral infection that still had healthy or even rising levels of gene expression in the host cell, i.e. the gene is not switched off but the protein is being destroyed instead. This analysis correctly identified all the ones we know about in adenovirus, and are known to be functionally important, and identified some new ones, one of which we were able to verify as an exciting new target. We think this will be useful to study well-understood viruses in order to see if they are targeting host proteins for destruction, and for the study of animal, plant, insect viruses in their normal non-human host, especially if you want to examine zoonotic viruses such as the ones that recently jumped from bats to humans in the Middle East. Anyway, hope you find it interesting. Best wishes, David. And he gives a link to a Nature Methods paper.
It's very cool. I mean, basically, if I get this right, when you, say, do total sequencing of, say, transcripts from an infected cell, you depend on databases to find all the encoded proteins. But what they're saying here is if you don't always have that, you could at the same time sequence all the proteins by mass spec and get both data and be able to see what's going on, right?
Mm-hmm. This it reminded me a little bit of the paper that we did, was it last week or 2 weeks ago, on hepatitis B, where in order to do their studies in, what was it, a tree shrew?
Right,
Yeah.
the tupaya.
Okay,
Yeah, that's right.
the tupaya, where they didn't have a database.
Exactly.
They did deep sequencing on the tree shrew and basically deduced a proteome from the transcriptome.
Exactly. Okay. Right. They had gotten the sequence of the protein pulled down by the Hep B protein, and they needed the transcriptome to find out what it was, what the rest of the protein was. Yeah, so they just did that. I think it's the same idea here. You get the proteome and the transcriptome at the same time, and then you can use the information together instead of having to search the database. And if it happens that your gene is not in the database, you're not going to see it. It's a pretty cool approach, I think.
Yeah.
David is in the University of Bristol. Thank you, David. And don't worry about self-promotion. It's virology.
That's how we find stuff out.
Yeah.
That's right.
It's a nice paper.
Yeah.
It is.
Kathy, you are with Mary.
Okay. Hi, I had a question. I was listening to Bats, Elephants, and A's, TWiV 10. I got everything about the elephants, but I didn't really get about the bats and the AIDS. What were the main points of that, and why are we so concerned about it? What were the overviews of those topics? Thank you.
Alan, doesn't this sound like one of those automated comments that get put everywhere? I mean, is it a real question?
I think this is a real question.
Okay.
Yeah, you're right. It's phrased generally enough, and it's a short enough note that it sounds almost like one of those I found your blog very interesting type of comments.
Yes, and then they link to Coach Leatherwear or something.
Right.
Well, she didn't link to anything. Bats, Elephants, and A's, TWiV 10. Oh, look at this, Alan. Hosts Vincent and Dixon. Guest Alan Dove.
Yes.
I think this is your first appearance.
I think it may be, yes. I think it is.
December 4th, 2008, 4 years ago.
Wow.
Vincent, Dick, and Alan chat about reconstruction of a bat SARS-like coronavirus, herpes viruses that are killing elephants in zoos, and a plan to eradicate AIDS in 10 years.
Yeah, there was no connection between the bats and AIDS.
No.
There were three different topics.
Pretty straightforward. That would make Alan next with Blaine.
Yes. Blaine writes, "Hello there, Vincent and Dixon and colleagues. I'm a biology instructor at a college in northern Alberta, and I've absolutely loved your podcasts, This Week in Parasitism and This Week in Virology, as they have helped me learn new and wonderful things. Now that I've given you some glowing praise, will you please read my questions in your next podcast?" Well put, Blaine, well put. My first question is relevant to both TWiP and TWiV, so I sent it to both podcast emails in hopes that there is someone out there that may have an answer or two. First, I've been thinking a lot about phage therapy, so my question is this: can we use these viruses in some sort of therapy analogous to phage therapy? Which
viruses? Yeah.
I've recently learned that the protist Trichomonas can sometimes harbor a virus that can make the disease more severe. Clearly, we don't want to encourage these viruses. I guess animal viruses in it.
So Vincent has two links, and for the phage therapy one, you should see TWiM number 6. And TWiM number 6 also has a transcript. So I was able to quickly look through that this morning, and it's really a nice description about phage therapy. So that's worth either listening to or looking at.
Kathy did the homework for this episode.
I did some of the homework. And
I think he was asking about phage therapy, right?
Right,
but
exactly.
Can we use these viruses? Was a little. I didn't.
And he talks about analogous to phage therapy.
Yeah.
It's almost like he's asking whether there are protozoan viruses or something like that you could use to treat other diseases.
Oh,
but
maybe.
I think that's the question.
Yes.
Okay.
Yes.
Well, those
And
protozoan viruses actually don't kill their hosts. So the trichomonas viruses, for example, are non-cytopathic. So I don't think that would be a good approach. But if you could find ones that killed them, yeah, you could. But
or if you could modify a non-lethal virus for the parasite into a lethal one.
Yeah.
Right. And Vincent also has the link to TWiP number 47. There was no transcript for that, and I didn't have time to listen to it. So
yeah, on TWiP 47, we talked about these trichomonas viruses. They're very interesting. They're double-stranded, real-like viruses, and Max Niebert works on them. And what he found was that they exacerbate trichomonas infections because the double-stranded RNA is sensed by innate sensors, and you get an inflammatory response. So it makes the infection worse by having these viruses present. They did a really nice piece of work there where they looked at that. I think you know someone, Rich, that works on this as well, right? I think one of the authors on that paper, he had met you at ASV or something like that.
I'd have to have a look at that. I don't know.
Okay.
So, I wonder if you can use Mimi virus to cure amoeba diseases. Or something like that,
Yeah. Well, sure. Why not?
Yeah. All right. Second question. I've been learning about herpes virus and had a basic question about herpes virus life cycles that I'm unable to find a sufficient answer to. How do these viruses go latent? Do they incorporate themselves into host genomic DNA like retroviruses, or is it by some other mechanism? Thanks a bunch for all the work you put into these podcasts, Blaine.
Some other mechanism. The genome circularizes. It apparently becomes chromatinized. That is, it acquires histones and stuff from the host, and it exists as a circle that is not a circular DNA molecule that is not covalently connected with the host chromosome, persisting in the nucleus, and just sits there more or less dormant, depending on exactly which herpes virus you're talking about.
Right. Okay.
And in general,
Yes.
the question, how do these viruses go latent, is sort of the $64,000 question.
Yes.
Yeah.
Not only how do they go latent, but how do they then reactivate? That's the $64,000 question. Yes, that's
The
correct.
reason you're not able to find a sufficient answer to that whole thing is that there isn't one yet.
Actually, it's worth a lot more than $64,000.
Well, yeah. But game show was when it was $64,000.
It's metaphorical.
Right.
Next one is from Steve, who is our transcriber. Hi, attaches the transcript for TWiV 206 viral turducken. I put this in here to show how much Steve has been doing for us.
Yeah.
Now, instead of transcribing the whole thing, he just did the part where we talk about the papers, which I think is fine.
That's fine.
He doesn't have the banter at the beginning. He doesn't have the email, but that's
fine.
He says if you prefer to have the whole podcast transcribed, just let me know and I'll get back to the rock pile on this one. No, it's okay.
No.
Anyway, my sympathies to Kathy. Sometimes the long-in-the-tooth dogs can talk and talk and talk. Actually, really informative for me. Thanks. Do we talk a lot on that? Is that what he's saying, Kathy?
I think yeah. I'll let Kathy answer that.
I think it might not have been the TWiV. Well, it might have been the TWiV 206 one, but there was another one where you guys talked for a while about are viruses alive, and finally I said, "Don't you want to know what I think?"
Maybe it was 206, right?
Well, no, that was a different one. But
Was that?
sometimes I do feel like it's hard to get a word in edgewise. But mostly it's because you're saying intelligent things, and I don't have to add to it, so that's okay.
He sent also a transcript of how to read a science paper, TWiV 169, which is really cool. So that's up as well. Okay. We appreciate that, Steve. Who? Let's see. I'm in New York,
you.
so that must be Rich Condit.
You read that one?
Oh, I read Steve.
Yes.
Now you're reading Andreas.
I can't even remember who read the last email.
That's bad. Me too.
Andreas writes, Dear Vincent, I am an avid reader of TWiV since some time, and I believe the following story would be in your scope. I worked for some time on host factor targeting, broad-range antivirals. Bottom line, we identified a physiochemical mode of action applying an FDA-approved compound acting antiviral against a broad range of pathogens. Understanding this mode of action enables how to develop better, more targeted compounds, opening a completely new field of antiviral development. It's pretty much the first time a host mechanism is targeted which has a significant benefit of not being virus-specific, shared pathways. There are also other nice aspects like synergy with compounds that could not be applied until now since they are too toxic in their effective range, but combining two different modes of action with the same target significantly limits toxicity. I hope the readership of your blog would be interested in such a finding. The paper was very recently published in PLOS Pathogens, and we have a link to it. I hope you find the paper interesting and worth to be spread. Please contact me anytime if you have questions. Thanks and all the best, Andreas. And he has a P.S. I'm not active in academia anymore and thereby do not want to hijack your blog for my career. However, I truly believe that our work will enable completely new ways of developing antivirals, something we desperately need. The more people in the field are aware of it, the better. So I didn't look at this in detail. You did, Vincent. Is that right?
This is cool. What they did is they screened a library of FDA-approved compounds, and they found one that inhibits, it basically inhibits part of the endocytic pathway. So it targets acidic endosomes. And it inhibits a variety of viruses that require those for uptake, like I think influenza and rhinoviruses. And since this is approved by FDA and it can be apparently tolerated at very high levels by people, you know, it's something that might be useful, could go into clinical trials.
Mm-hmm. Yeah, I've seen this approach before of with some new agent screening or some agent where it has not been tested, screening already FDA-approved drugs, because there's quite a few of them, which it's a brilliant approach, because then you don't have to go through a lot of the preclinical testing's already been done.
Yeah.
You can actually buy that library off the shelf.
Yeah, right.
So anybody out there who's thinking, gee, you know, wouldn't it be great if we had a drug to do X, but nobody's looked for it? This is absolutely the very first place you look for whatever activity you think would be useful.
Right. Now, they find that it's active against, as I say, flu and rhino, and these, of course, are respiratory infections. So I just wonder if an orally administered drug would work, or if you would have to target it to the respiratory tract by an inhaler, you know. So I don't know what. So, Alan, would you have to do an animal model first before you went into people?
Since it's already an approved compound, I think you can go directly into people. I mean, in theory, doctors can, if it's FDA approved, doctors can prescribe it for any indication off-label. You just can't sell it for that.
Right.
So you should be able to set up a clinical trial, go straight to clinic, because it's got fully characterized toxicity profiles and all that sort of thing, and side effects.
Yeah.
Just get your patients and figure out how you're going to design your study. It wouldn't be cheap, of course, because clinical trials are the expensive part anyway. But you'd be able to skip past all the preclinical stuff that you'd normally have to do.
Now, if the drug is approved orally, would you have to, and you wanted to say try it by a nasal spray, would you have to do something else?
I think you'd have to. You'd need to do some kind of additional work to prove that that's an acceptable delivery mechanism.
I guess you could see if. I mean, I'm just wondering if orally administered would work for a respiratory. I mean, you know, Tamiflu works for influenza, so it could. I just don't know. For this specific one, if it would or not. Anyway, it's pretty cool.
So I just wanted to point out that some of this was. Some of the authors are from the United States, but the senior author is Urs Graber from Zurich, Switzerland, who's a really good adenovirologist and very active in virus entry and things having to do with endosomes. So
Right.
Just wanted to point out that scientists.
It's international collaboration, right?
Right. Right.
Okay. Rich, did you read that one?
Yep, I read
Okay,
that one.
so Kathy,
That I remember.
You're back to Jim, Kathy.
Right. I've gotten to read both of Jim's. So, Vincent, I saw this New England Journal of Medicine article about the response after Sandy. At the article's end is a reference to the National Center for Disaster Preparedness, Mailman School of Public Health, Columbia University, New York, which I've not noticed anywhere before. I emailed one of the article's authors, Dr. Redliner, and the center to see if they had heard about programs like Sahana Eden. And I got this interesting response from the center that includes a webinar tomorrow, the 27th, which was 27th of November. And then he includes this email, which says, "Hi, Jim. Thanks for touching base with us about online central data collection systems. Yes, we're interested in this as well and have been looking into best practices. Sahana Eden and Usahidi seem to be the most widely used, but there are always new developments. At the current time, the Columbia Regional Learning Center doesn't provide actual software systems for this purpose. That said, we do have some online courses pertaining to usage of social media during disasters, which can be found here." And then he mentions an upcoming webinar, which is already passed. So then back to Jim's letter. I see that there's a New York Distance Learning Association, which is news. Also, through that site, this list of PDFs dealing with such health-related topics as vaccines and dengue. You probably deal with these places and sources because they are local, but they might be of interest to your listeners, too. I was particularly pleased to get information about Usahidi, another means of dealing with disasters. The COM 2302 class didn't look too useful. Jim. So, and then Alan is adding a note here that FEMA also offers a slew of disaster preparedness response mitigation courses. But I did look at these two links, and I think the main way to summarize it is sort of the usage of social network data during disasters.
That's kind of crowdsourcing disasters.
Right.
It's amazing.
Yeah. It's something. Yeah.
Crowdsourcing disasters in a managed fashion. It's just incredible what's happening. This stuff has a life of its own. Mm-hmm. It's amazing.
Yeah, because these data are being generated. The FEMA link that I provided is that's not data specific. That's a whole bunch of stuff. How to everything from how to manage a hazmat spill if you're a professional in that field to they have courses for how to put together your home emergency kit for when the lights go out. But these social media things that people are looking at now, and this is still very embryonic, I think. The data are being generated. People are tweeting and putting Facebook updates and hey, my electricity went out. Hey, a tree just fell on my neighbor. That kind of thing. And the ability to tap into that could be immensely useful.
Mm-hmm.
Yep. Boy, Skype has settled down, hasn't it?
Yes.
Good. And,
Good.
I think it's your turn, Alan.
Okay. Marion writes, first, regarding the extended discussion of the usefulness of the SARS-CoV exon mutation for attenuation of SARS virus in hopes of future vaccines. Just an opinion, but the success and extremely high safety of the subunit HBV and HPV vaccines suggests that most future vaccines will be engineered, especially considering the climate caused by the anti-vaccine wackaloons. Finding correlates of protection will be more important than correlates of attenuation.
Right.
Second, regarding Matt's pick, virus-associated pyramids, slide number 11. Having been educated at Rockefeller University, one of the founding places of cell biology, and being taught by several Nobel laureates who discovered organelles, it was drilled into me that EM is highly prone to artifacts. You could look at sodium chloride and see seven-faced pyramidal structure. EM data alone are meaningless. It must be corroborated with data from other methods. Moreover, it was a color-enhanced EM. Electrons are not in color. Many people did not understand that. While I understand that his pick was aesthetic, it should not have been accompanied by such it should have been accompanied by such warnings.
All right.
They're pretty pictures.
They're nice.
Yes, they're pretty pictures.
Yeah, false coloring helps us to see things, and yeah, we can try and point that out.
And it was an aesthetic link, yes.
Yes.
All right. It's my turn, right?
Yep.
This is from Winkler. He writes about the TWiV with John Udell. Is there any way to compare the productivity of NIH research model with the university research model? Now, if you remember from the Udell TWiV, he was advocating an NIH model, more extensive NIH model in universities, because you don't know, not everyone has to be a PI. You could be a staff scientist, for example, which they have a lot of at NIH. So I emailed this to John, and he said 2 thoughts. 1, yes, in principle, can take some objective measure, number of citations, and divide it by money, funding, but both are problematical. We all know difficulties of measuring true impact, so citation number is an indirect proxy. Money funding is surprisingly difficult to nail down since universities have Byzantine financing and with many subsidies for supporting research and typically can't provide real costs. On the other hand, devoted research institutes, limited in number, particularly small ones, exactly know their costs. 2, as members and stewards of a profession, we would be completely stupid not to factor in some level of investigator happiness in improving the current system or trying to rationally design an optimal system. If System A is demonstrably more efficient, but the investigators are miserable, is this really preferable? System A will also over time drive out/repel more talented creative individuals, and the productivity is and the lost productivity is incalculable. Well, there you go. You could do it. I don't think anyone's done it. I'm not aware of it.
Yeah, and I would add that this whole idea of productivity measures, they can be incredibly useful in certain contexts, but they're also a bit of a trap if you're not very, very careful about how you proceed.
Sure.
Because you can come up with a number, and as soon as you come up with a number, then everything non-quantifiable suddenly looks irrelevant. And depending on what that number encompasses, it may lead you down a really, really evil path. So I know there's been this huge rush to quantify the efficiency of our healthcare system, and that's had some benefits, but it also has been incredibly corrosive in a lot of ways. Where you say, well, gee, we could get more productivity in units processed out of this doctor if they only see patients for 10 minutes.
Right.
So now we're going to have this doctor seeing patients in 10-minute intervals. Of course, that's really not going to encompass all that needs to be done in the context of medical care, but you can't quantify a lot of the rest of that, so it falls by the wayside.
Yep.
And to some extent, this is already happening in research, where we've got, the Church of the Holy Impact Factor, and I think that's not been a positive influence overall.
Yeah. And he says that his second point is that even if you could make such comparisons, you got to just see if people are happy. And if they're unhappy in the university system with the PI on top and everyone else below, then you got to factor that into it. So, and a lot of people are not happy these days. Right?
Yeah.
All right. I read that, right. So that brings us to Rich Condit.
Steve writes, "Hi, I have only listened to about 25 TWiV podcasts. If it is not a regular recurring segment, I encourage you to have a host periodically focus on the statistical methods and analysis in various studies. Statistics is an important broadly applied and sometimes misapplied general science tool that could benefit from more in-depth critical discussion. Also, in doing various topic searches in the TWiV archives, I find it difficult to get good search results. In addition to listing the podcast number and title fields, it would be nice if the description field was also shown. These more clearly express the content covered in each podcast." So with respect to the statistical methods, we've had a guest on a couple of times, Michael Walsh, and I think his first major appearance was in TWiV 169, titled "Epidemiology Causes Conclusions."
Yep.
And we had him on, I think, a couple of times since then. We can probably search for his name and figure that out. But he's right. The statistics are an important part of this. It's something where, personally, I'm pretty lame, and it's nice to have an expert on.
Yeah, we often don't bring up the statistics, but we talk in such broad strokes about findings that, I don't think we would do that, right?
Right.
Yeah, I think if we have, particularly if we have a paper that's really heavy in statistics, we'd probably want to bring on one of the authors, or at least maybe bring Michael back and and have have somebody weigh in if we're really going to be wading into the math.
I think basically when we discussed the H5N1 sero surveys, we brought Michael on to help us with it, right?
Yeah,
that was that.
And he was on. Yeah, he was on again in TWiV 173, going to bat for flu research.
Right.
Who makes up these titles anyway?
Alan Dove.
I can't imagine.
Alan Dove.
Somebody with some neurological defect.
Yeah, that's it.
All right. So we're back to Ann Arbor, Michigan.
Josh writes, Vincent, it looks like the powers that be did a pretty good job in choosing the flu strains for this year's seasonal 2012-2013 vaccine. See antigenic characterization of CDC link, and he sends the link. Outpatient visits are up compared to the pandemic season, but it seems too early to tell as of yet. Keep up the great work. Josh in Georgia. So when I first went to the site, I was kind of overwhelmed and couldn't quite figure out where he was getting the information that the vaccine guys got it right, or the vaccine group that decided what to put in got it right. But if you go down a ways, where is it now here? Antigenic characterization. Yeah, he says that. Okay, so for the influenza A, first for the 2009 H1N1, all 10 H1N1 viruses that they tested so far this year have been just like what's in the vaccine, evidently, if I interpret that right. And for the H3N2, 99.1% of them have been like what's in the Northern Hemisphere influenza vaccine. And for the influenza B, 69% of them have been characterized as the influenza B component, and 31% have been from a different lineage of B viruses. So, so they have done pretty well, it sounds like.
Pretty good, yeah.
Very good.
Yeah.
So do I understand it correctly that after a big burst onto the scene,
yes,
the 2009 H1N1 has taken kind of a back seat?
Yeah, it's very interesting.
It's very interesting.
H3N2, which has been around since 1968, is still going strong. It's very interesting.
I think maybe the TWiV bump for H1N1 just didn't last.
It did not. We talked about it an awful lot, didn't we?
We did, and it was very prevalent that year.
But I think people probably went out and got their vaccines.
Yeah.
Yeah. So what you're referring to is the fact that in compiling these data that I just recounted, the CDC characterized 351 influenza viruses. 10 were the H1N1, 226 were the H3N2, and 115 were the influenza B.
Yeah, that particular week, yeah. It's very cool. This site is great for seeing what's happening every week with flu in the season. Check it out. All right. I think we are up to Alan Dove.
Yes. Eva writes, Dear Vincent, I really enjoy your podcasts. In the last episode of TWiM about the spores, you mentioned that you have had an argument about whether the viruses are living or not. It seems a very interesting topic to me, and it will be great if you could send me a link to that podcast. Thank you in advance. I think that's not a single episode that we would point to. Who did that?
Rich Condit.
That wasn't me.
No?
It wasn't me.
Okay.
We have talked this one to death. I looked back.
Why it set off an alarm?
I looked back and found discussions of this in TWiV 48, minutes 5025 to 5350. In TWiV 59, minutes 5530 to 10510. That was a long discussion. And Kathy found a couple of more recent ones. TWiV 209, an hour and 7 minutes to an hour and 15 minutes in response to a listener email. And we also, TWiV 59 was where we put up the survey, which that link is now dead, I think.
Is that right?
Yeah, I clicked on it from TWiV 59 and didn't go.
Right.
I think it said it was
I have
dead.
to fix it because it's the poll is still up there. Let me
Okay.
fix link. to poll. Okay. It's there. There are actually quite a few people who have web pages are not responding. Come on. Why not? Let me just tell you how many people have answered. So if you go to virology.ws, there's a link at the top that says, "Are viruses alive?" And you're right. It's dead. It's gone.
How does that happen?
Survey Monkey. What happened? I probably just have to fix the link. Well, there's a lot of discussion comments there. Yeah, you should check them out. I'll fix the link, though, I'm sure.
The virus killed your Survey Monkey.
Ooh, now my whole sheet is down. Let me re-sign in. All right. To my Google thingy. Okay, here we go. I think we'll do maybe one more.
One more?
Does that sound like a plan?
Sure.
Who did the last one? I forgot already.
I did the last one.
So that brings us to me.
Brings it to you.
All right,
We're
the
on
last
to
one
Doug.
was Joss. The next one is Eva, and then Doug. Okay. TWiV folks, I've been listening on and off for the better part of a year, and with the successful acquisition of a functioning iPod, you guys have filled my lonely hours on the bus as I transit through the Appalachians to and from graduate school. Anyhow, being a bit obsessive-compulsive, I wanted to start TWiV from the beginning, and after being heartily entertained by a rant from Dr. Racaniello and Dr. Dove about stomach viruses being blamed for all GI ailments, this headline immediately made me think of you guys. Clinton cancels Middle East trip over illness. Probably everyone knows that Hillary Clinton had some gastroenteritis recently and had to cancel a trip. So my questions, in accordance with the Vincent Alanian standards of proof for virus-caused GI distress. One word, Vincent Alanian, similar to Koch's postulates, but tougher. One, is there a reasonable expectation for a Secretary of State to perhaps have not a PCR machine in her bedroom for virus ID, as you've suggested, but at least some lackey in the Department of State who can run a sample of some body fluid tissue to a person capable of confirming the presence absent of a Norwalk or norovirus, rotavirus? If yes to the above, when does this capability become available to cabinet officials? Are there any historical precedents for virus-induced illnesses canceling high-level, if futile, summits? Cancels a lot of sporting events, I know that. You always hear of some player who's got a stomach virus and can't play. I don't think they even diagnosed her illness. I think you get gastroenteritis, vomiting, diarrhea in the winter, and it's either a noro or a rota or any of a number of other viruses that could cause it, right? Because there's nothing you can do anyway except give them fluids.
Yep.
Right.
Now, in her case, they didn't give her enough fluids. She got dehydrated and she fell and she hit her head and got a concussion, I understand. So if you have these winter vomiting diseases, you got to take a lot of fluids.
Or if you have any kind of gastrointestinal upset, you don't have to diagnose it to know that you're losing fluids.
Yeah, that's the treatment, restoring fluids.
As for historical precedents, George W. probably wishes they had canceled a summit when he was sick.
Yes.
He managed to embarrass himself by getting sick at the dinner table, as I recall, or at some discussion.
Wasn't it on Japan's prime minister?
Yeah.
Oh, my gosh, I remember that. Yeah. Thanks for giving me an excuse to blow 10 minutes off from studying for finals oral candidacy. You guys rock. The big whale. He signs his email. Well, you didn't really blow 10 minutes off. You learned something.
Right.
Right? Did you and I rant, Alan? I can't imagine.
Us ranting?
I don't believe it.
No.
I ranted to my graduate virology class. Well, I pretty much always do, and I have quotes and things about how there is no such thing as the stomach flu.
Right.
And shortly after that, in one of the dorms here, there was an outbreak of norovirus, and the headline in the Michigan Daily, the student paper, was something about stomach flu hits the dorm, and so my students rushed to show me this. Just went, "Ah!"
I used to have a few tirades that when I taught the journalism course at NYU, I had a few little tirades like that I would go on.
Yeah.
Well, we did pretty well. We only have 1, 2, 3, 2 left, actually.
Really?
Yeah.
So this is all of our email.
That's it. I think a few came in this week. I didn't put them on, but yeah, we caught up at the end of the year. How about that?
Great.
Awesome.
Let's do a couple of picks. Let's start with Rich. What do you have for us this week?
So this is a video from an organization called the Planetary Collective. This my wife discovered and sent to me. It's about a phenomenon called the overview effect, and it's a nice little video that describes basically, I would call it a fundamentally spiritual experience that is described by numerous astronauts. Let me define spiritual here. This is from I'm using the Dalai Lama for my definition of spirituality. Qualities of the human spirit such as love and compassion, patience, tolerance, forgiveness, contentment, a sense of responsibility, a sense of harmony, which bring happiness to both self and others. This is as distinct from religion. So apparently it's not surprising and not uncommon for people who get shot into space and who see the planet from a distance to be experience really transformative awe and get a new perspective on the unity of things and how the planet is, sort of the Gaia experience. And this is a video that summarizes that. I got to make a little bit of a disclaimer here. The prime mover behind this is a guy named Frank White, who originally formulated this into a book that he wrote. And this morphs into an overview institute where people want to communicate this sense of unity, if you like, and how we have to take care of the whole planet to others. So I suppose you could accuse Mr. White of a little self-promotion for this, but I'm gonna prefer to think that he actually has altruistic motives. So I think it's worth looking at. It's interesting to hear the astronauts talk about their experience. But I would also what I liked about it is that it communicates how one can, through science, and in this case, exploration of space, get a sense of how integrated not just life, but the universe is. And everything is related to everything else and sort of transcend all the petty day-to-day stuff that we deal with. I would say that you don't have to, although getting shot into space would probably help induce that sort of feeling, I can get it from looking at a microscope. So there are different ways to approach this.
I was going to say, a plaque assay does it for me.
Right, exactly.
But I think I would say that that absolutely this, I think somebody commented that the single most jaw-dropping photograph ever taken was shot by an amateur through the window of a moving vehicle.
Yeah.
And it was that Earthrise photo that we've all seen. It's absolutely amazing that when you look at that, you think what it entails. Just wow.
Yeah, it's pretty cool.
The nicest interview on this thing is with Ed Mitchell, who was one of the guys who walked on the moon, where he talks about having an hour or so of time when he was in the command module, which was rotating so that every few minutes he had go by him the moon, the earth, the stars, just cycling by, and what a transformative experience that was for him. Enough so that when he came back, he had to kind of study this and try and understand what had happened to him. Okay, he's very interesting on the topic. At any rate, if we can communicate that to everybody, I'm all for it.
It's cool.
Cool.
Mm-hmm.
Very nice. Alan, what do you have for us?
Oh, well, after that note, I have something completely and utterly silly, which I found very entertaining. This is a publication called Notes Towards the Complete Works of Shakespeare. And the authors on this are Elmo, Gum, Heather, Holly, Mistletoe, and Rowan. They are Sulawesi crested macaques from the Paignton Zoo Environmental Park in the UK. And as the title suggests, this is what these particular macaques have come up with when given a typewriter and allowed to start working on the complete works of Shakespeare. So this is testing a very old hypothesis. And you can download this.
Yeah.
You can get a copy of their notes.
It's all single letters.
Yes, it starts off FF and then.
Alan, when I first looked at this, I thought, my first reaction was, this is something serious.
Yeah.
Picking Shakespeare, this is really weird because it's got this nice picture of a sort of a battered leather-bound thing. And then I looked at the authors.
Yes.
Yeah.
Elmo, Gum, Heather, Mistletoe, etc.
It's a quick read.
This is yeah.
And down at the bottom, they have further documentation, and you can see pictures of the authors.
Yeah,
about
great.
the authors, right? This is hilarious.
That's pretty cool. So it was a typewriter they gave them, is that right?
It was a computer terminal, I think.
So they could actually see what they were doing?
I am not sure.
Yeah.
They may very well have just sat on the keyboard.
Yeah, right.
Yeah.
That's great. Nice. Thank
They
you.
got a good start.
Yeah, it's good.
Yeah.
Some might argue it's better. Kathy, what do you have?
So I had a couple of picks, mostly about correlation versus causation. So the first is a picture from one of those picture sharing sites about did AVAs cause the housing bubble? So there's a graph of the housing price index over time and babies named Ava. Or is global warming a hoax propagated by scientists? So they have the National Science Foundation R&D budget and the average global temperature. So there's several of those. And then
I love the mountain range.
The mountain range one is really good.
That's awesome.
And then this one won't be accessible to everybody. It's not an open access paper, but I think I've mentioned my colleague who's Swedish is on sabbatical right now in Sweden, and she sent us this link to a paper in the New England Journal of Medicine about chocolate consumption, cognitive function, and Nobel laureates. And even if you can't get to the paper, I noticed that you can at least get to one of the key figures where they just kind of show the correlation between country's annual per capita chocolate consumption and the number of Nobel laureates per 10 million population. So it kind of implies that, maybe if you eat more chocolate, you might get the Nobel Prize.
And speaking of statistics, the correlation here is amazing. The p-value is quite low.
Yeah.
Right. And then maybe we can include this one that Alan sent too, which is a correlation between autism and organic food sales, just to show you that, you can kind of show correlations with things that aren't necessarily showing causality. So that's what I have for today.
So what's the moral of this, Kathy?
The moral is that just because things have the same shape of graph or are in the same place or same time doesn't mean that they're necessarily related.
Right.
I thought the moral was I should eat more chocolate.
Yeah, you could do that if you
Yes, that too.
Yeah.
want. Yeah, we talk about this a lot, right?
Yeah.
Always, because every time when you tie your shoes and it rains, doesn't mean that you caused the rain by tying your shoes.
I had actually seen this one about autism and organic food sales, and I tried to look up the source here, which is listed under the graph, the Organic Trade Association, blah, 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 blah. But I couldn't actually find my way back to the original graph. I was interested in how it was used.
Yeah.
All right.
Cool. Thank you, Kathy. And that brings me up. My pick was inspired a couple of weeks ago. I was giving a seminar at Albert Einstein here in New York, and a young lady in the third row had a T-shirt on with the periodic elements fluorine, uranium, carbon, and potassium. And
Uh-huh.
Right?
Okay.
And I was looking at it and go, wow, where did you get that? So I looked around and I found a site called Periodically Inspired, which in fact doesn't have that particular T-shirt on it.
I was
But
going to ask.
They're T-shirts and mugs with other combinations of element symbols. One of my favorites is OMG, or Oxygen and Magnesium.
Mm-hmm.
It's a pretty cool one. Nerdy, genius. And they have shirts and stuff. So it's pretty cool.
I saw one last night at a friend's house sitting on their dining room table. They hadn't wrapped it up for the recipient yet, and it was BACON. I can't remember quite how it divided out into the
it's good. Yeah.
elements, but yeah, it was good.
Yeah, so if you're looking for a gift, although probably wouldn't be ready in time for Xmas, there it goes.
Yeah.
Periodically Inspired. We have a two listener picks, one from Mark. Mark is our friend from the Drobo days. Some of you may remember. Dear TWiV, on your podcasts, you talk often about various aspects of peer review and scientific publication. I recently found this video that presents Nature's process. It is geared to a lay audience.
That's Nature with a capital N.
That's
it's
right.
Not Nature's process.
Yes.
Yeah.
So check that out. That's really good. And the other one is from Ricardo, our friend in Portugal, who writes, "Hello, Vincent, and growing number of friends." Can always use more friends. Here is a nice post about going viral. And it's called Things Don't Go Viral. Let me get it up here. Except viruses. Just go look at it. It's pretty funny. You've heard the stories about viral videos that receive millions of views. One part of those is not true. Things don't go viral the way that viruses do. This is a little spiel about that. So thank you, Ricardo, for that. And that will bring us to the end of the second to the last, the penultimate TWiV for 2012, which you will find on iTunes at twiv.tv and at microbeworld.org/TWiV. If you do like us, go over to iTunes, subscribe, and leave a comment or a rating. That helps us to remain on the front page, which we're very successful at doing, and that's how more people discover us. Do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun, and as always, I learned a lot.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
My pleasure. Always a good time.
And Alan Dove is at alandove.com and on Twitter. And thank you, Alan.
Always a pleasure.
And be sure to check out Alan's blog post on the end of the world today.
Ah, yes.
I need to look at that.
Okay, good.
I'm Vincent Racaniello, and you can find me at virology.ws. For those of you that celebrate Christmas, we hope you have a merry one. And we'll be back at the end of next week, which is still 2012. And we'll be having our annual Stories of the Year in Virology. So don't miss that one. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.